Hi, this is Dr. Kurt Woeller from Integrative Medicine Academy. The title of this video is Autism, Clostridia bacteria, and its link to mitochondrial damage. So one of the things we know in the world of autism and autism spectrum disorders in general is that there's many things that contribute to it. So many of these individuals, whether it's a child, a teenager, an adult, have a variety of nutritional imbalances. They've got dietary problems. They usually have gastrointestinal issues as well. They're also very susceptible to environmental chemicals. Glyphosate, organophosphates, for example, heavy metals can certainly play a role. Candida, bacterial toxins also seem to be contributing factors to many of these individuals' issues because all of these things compound and can cause problems in language development, cognition, awareness, eye contact, etc. And one of the things that has been appreciated, perhaps maybe overlooked to some degree, is mitochondria. Now, mitochondria are fundamental to our body, pretty much every cell of our body except red blood cells, in the role that they play in energy production. We're going to talk about that, but there's some other things to consider when it comes to mitochondrial issues, particularly in relationship to Clostridia bacteria. Now, before we get into that, I just want to show you a few slides. This is actually a paper on dopamine toxicity. Now, this particular paper is mostly referencing dopamine issues related to Parkinson's disease. And so I'm not insinuating somehow that autistic individuals have Parkinson's, but there is an overlap in some individuals with regards to dopamine issues. You'll notice this complex 1 and complex 3. That's actually part of the electron transport chain of the mitochondria, which I'll show you here shortly. Isocitrate dehydrogenase is actually part of the Krebs cycle. And these are two interesting areas within mitochondrial function that become compromised, at least in one avenue, with regards to dopamine toxicity through a chemical called aminochrome. Now, there's a lot of things that happen downstream from that, but the end result is mitochondrial dysfunction leads to an energy production problem and can lead to neurotoxicity or neurological metabolic imbalances. So one of the things within the nervous system is that when we have high levels of dopamine, it creates a compound called dopamine O-quinone. And dopamine O-quinone ultimately can lead to superoxide radical formation. These are oxygen species that create reactive oxygen species generation and can lead to neurodegeneration. Now, typically we should have enough glutathione, but often autistic individuals are deficient in glutathione, and it leaves the cell vulnerable to oxidative stress. It can also lead to something called apoptosis, which is a mechanism where the cell essentially commits suicide because it becomes damaged. So there's a lot of different biochemical pathways that are linked to this, but ultimately dopamine O-quinone can create something called aminochrome. And aminochrome, as I just showed you, has some relations to mitochondrial problems. Now, in Parkinson's, aminochrome metabolism eventually forms neuromelanin. And so we get this pigmentation that occurs in the areas of the brain in Parkinson's, for example, like the substantia nigra. I'm not, again, insinuating somehow that this is occurring in autistic individuals, but one of the things that is occurring, is that autism as a exploding diagnosis is really a relatively recent event. I mean, we're talking probably in the past 10 to 15 years. And we don't know what the future holds, but what we do know when we look at the underlying biology, underlying molecular mechanisms, underlying biochemistries, we can see relationships here where dopamine doesn't necessarily have to generate a neuromelanin effect. It can just generate reactive oxygen species that can compromise the autistic individual's brain and nervous system function. So aminochrome is a very important chemical to remember. And so ultimately, if we have aminochrome buildup, we're going to get ATP depletion because aminochrome then creates problems, or the metabolism creates problems that affect the mitochondria within the Krebs cycle and the electron transport chain. So if we think of metabolic demand, there's no bigger demand in our body than the brain or nervous system. And probably you would say the cardiovascular system, maybe both are kind of running neck and neck, but both need a lot of ATP, the adenosine triphosphate that gets produced by our mitochondria. And when we have deficits of mitochondrial function in various organ systems, we're going to have dysfunction in those organs. And that could occur in the brain, that could occur in the liver, that could occur in the cardiovascular system, the musculoskeletal system, etc. And so there's various areas in the brain of an autistic individual, their frontal lobes, the hippocampus, the temporal lobe, cerebellum, that are often compromised. And so the efficiency at the mitochondrial level is key, right? Whether it's proteins, carbs, fats, ultimately these all get funneled down to help us make that cellular energy chemical called ATP. And so often the mitochondria is considered to be the energy factory of the cell. We make massive amounts of ATP at any given moment throughout our lives. And so the amount of ATP that's actually produced is almost incalculable, but the mitochondria, we know, are also vulnerable to different types of stressors, nutritional imbalances, different toxins. Those could be endogenous, exogenous, and other factors as well, genetics, for example. And so when you dive a little bit deeper into the biochemistry in looking at mitochondria, we know that the mitochondria have this outer membrane, they have an inner membrane, and then sitting within the matrix of the mitochondria is the Krebs cycle. And the Krebs cycle communicates with the electron transport chain via a couple different points. One is through the production of something called NADH, which is nicotinamide adenine dinucleotide, which is the reduced form, the hydrogenated form. And then it also communicates with the electron transport chain through FADH, which is a flavin adenine dinucleotide. And those both come from the Krebs cycle. And ultimately, it's to produce large amounts of ATP. Now, there are other functions of the mitochondria. Calcium signaling, they play a component in heme synthesis for hemoglobin production, for example, immune regulation. They help get rid of old damaged cells. Even steroid synthesis can come about from mitochondrial activity. So it's not just ATP energy production. It plays this major other role in regulation of cellular activity. Now, one of the things that's really critical to understand is how to utilize and interpret the organic acids test. This particular one comes from Mosaic Diagnostics, which was formerly Great Plains Laboratory. This is actually from an autistic child, where we can see high levels of lactic, high levels of pyruvic. So we've got some interference happening in the glycolytic or glucose pathway. We've got high levels within the Krebs cycle metabolites. And then we've even got an amino acid metabolite that's elevated. One of the other things that the organic acid test provides is insight into certain nutrients like CoQ10. CoQ10 plays a fundamental role in the electron transport chain for the transportation of electrons between the different complexes. Now, it's been known for years that individuals with autism are often compromised by the presence of certain Clostridia bacteria. And it's not just Clostridia difficile that would be driving an inflammatory bowel issue, but there are other compounds that different types of Clostridia produce. And so this particular one called HPHPA is very often present in autistic individuals, whether they're children, teenagers, or adults. This was a paper that came out years ago that was looking at this HPHPA marker detected through an organic acid test being shown to be much more prevalent in autistic boys than boys of a similar age but control, essentially, who are neurotypical. And so it's very common on organic acid testing. You have to do the Mosaic lab because they have the most robust Clostridia section. And we can see in this particular autistic individual, they have 3 of the 4 elevated. They've got the 4-hydroxyphenylacetic, the HPHPA, and the 4-creosol. And all of these are neurotoxins. They can all create problems in the brain. In fact, it's very common on organic acid testing to have at least one of these Clostridia markers present. About 1 in 4 organic acid tests you'll do will have at least one Clostridia marker elevated. It's usually HPHPA, but 4-creosol is also prevalent as well. And here's what's happening biochemically. In the presence of these bacterial compounds, these Clostridia bacterial compounds like HPHPA and 4-creosol, these things can interfere with an enzyme called dopamine beta hydroxylase. And dopamine beta hydroxylase is what converts dopamine to norepinephrine. And it doesn't mean that every single enzyme in the body or the brain is getting inhibited, but a certain percentage creates imbalances. And so when we inhibit the enzyme, we get an increase of dopamine. Now, if we remember that slide I just showed you, increases of dopamine can lead to that quinone type of product. And then from there, that quinone can become an aminochrome. And that aminochrome, via the body's attempt to try to metabolize it, can damage the mitochondria. So if we go back to this slide, here's our dopaminoquinone. This one isn't showing us the aminochrome compound, but we know that it gets produced. And so let's just say the aminochrome is sitting right here. Anytime we have a toxin, for example, right, that toxin needs to be neutralized by toxins internally, or excuse me, antitoxin chemicals in our cells. And so in the brain and nervous system, we've got glutathione, for example. And so with increasing levels of dopamine, we can get increased levels of dopaminoquinone, which means we can get increased levels of aminochrome, which over time means it could deplete glutathione. And that leads to the superoxide radical formation, neurodegeneration. Now that neurodegeneration effect occurs in part through dysregulation and dysfunction of the mitochondria in the nerve cell. So it really comes back to just understanding what's happening with dopamine and things that manipulate that. Well, we know that Clostridia, many of them can manipulate the dopamine levels in autistic individuals. And so therefore we have to understand that the organic acid testing is an absolute critical test in order to determine first if even the Clostridia markers are elevated. And they usually are. I'm not indicating that they're elevated in every single autistic individual, but you know, a good percentage, 40, 50%, will have at some point a Clostridia problem. So it cannot be ignored. So let's go back to our aminochrome. So we now know that the aminochrome comes from dopaminoquinone. Dopaminoquinone comes from dopamine. Excess dopamine that occurs in the nervous system because of the presence of a Clostridia toxin that's being produced in the gut could ultimately downstream from that lead to problems of our electron transport chain affecting complex 1 and 3 and the isocitrate dehydrogenase. Now, what's important about complex 1 and 3 and isocitrate dehydrogenase is these are critical steps or areas in both of these compartments of the mitochondria. So let's take a deeper look at where these things are. So isocitrate dehydrogenase, okay, is a critical step in sort of that first stage of the Krebs cycle. And it's very important for the production of alpha-ketogluteric acid. And alpha-ketogluteric acid becomes succinyl coenzyme A, for example. And there's a lot of chemistry involved. For example, succinyl coenzyme A is important for the production of heme. So heme could be hemoglobin to carry oxygen. Heme is also involved in cytochromes that are embedded in the electron transport chain of the mitochondria. Alpha-ketoglutarate is also important in the regulation of glutamate, and too much glutamate building up in the nervous system is toxic to nerve cells. So when we affect an enzyme like this, we're going to affect things that are happening downstream from there. In the electron transport chain, we've got complex 1. That is the main entry point into the electron transport chain from NADH. And we've got complex 3. Well, it turns out that we get maximal ATP production when we engage the electron transport chain at complex 1. So if we take out or compromise complex 1, we're going to compromise ATP production. Complex 3 is very important because this is the final step in the electron transfer to oxygen to make water. Now, sitting in the matrix down here is oxygen, right? So we've got a bunch of oxygen that's sitting in this space. Well, it just so happens that the electron transport chain, which is transferring or transporting electrons from one complex to the next to ultimately reduce oxygen to water, which helps in ATP production, if this is blocked and this is blocked because of something that is affecting it like aminochrome, we get electron leakage into the matrix of the mitochondria. Well, that becomes a problem because the interaction with electrons and oxygen that's not attached to one of these complexes generates oxygen radicals. And so the oxygen radical now itself is building up in the matrix, and it can then further damage all of these complexes, right? And that ultimately downstream leads to a depletion of ATP production. And so we get reduced ATP within that particular mitochondria. And if it's happening throughout the body, the impacts can be significant. If it's happening in the brain, well, we're going to have compromised cognition, attention, focusing, language abilities, etc. Now, we get into this information in very great detail in our Autism Mastery Course through Integrative Medicine Academy. This is a very extensive course on the biomedical intervention and aspects of autism. We have a new course starting Wednesday, May 31st, 2023. We generally have all of our courses available for purchase at any time. But this is a course that is fundamental to learning how to understand the complexities of autism and autism spectrum disorders and how to quickly intervene in many of these kids, or teenagers or adults who have a very complex problem. Now, one of the other courses we have through Integrated Medicine Academy is the Mitochondria Mastery Course. And this is all about mitochondria from its biochemistry, from its structure, its function, the genetics, and then other things that influence it, nutritional, toxins, other factors. So this is a very in-depth course if you are looking to get deep into mitochondria. What we do with the Autism Mastery Course is we do talk about mitochondrial issues that are related to autism. And some of the information from the Mitochondria Mastery Course is infused into the Autism Mastery Course in those areas that are related to mitochondrial issues. Now, we're talking in the Autism course more than just mitochondria. We get into diet, supplements, gut problems, methylation issues, folate imbalances, environmental toxins, mycotoxins, etc. So we cover a wide variety of details in the Autism Mastery Course, but we do pull some information from the Mitochondria Mastery Course to help you understand the in-depth nature of mitochondria and how it is related to many of the autistic situations or problems that we see in clinical practice. So for more information about the Autism Mastery Course, you can go to autismmasterycourse.com. You can also go to our main website at Integrative Medicine Academy, and that has information about our different courses. Each of our mastery courses has their own website, but you can also email us directly if you have any questions, and that email would be integrativemedicineacademy@gmail.com. Okay, so just reach out to us with any questions you have. Again, I'm Dr. Kurt Woeller for Integrative Medicine Academy. Thank you.